Hi, I am Bharadwaj Nandakumar. I'm an assistant scientist at Dr. Ganser Neurotechnology Laboratory at the Miami project. So it's been 1 year since I joined the Miami project, and today I'm going to talk to you about my PhD work under Dr. Karen Moxon at UC Davis, where we looked at the role of trunk sensorimotor cortex in supporting recovery of gait and balance after spinal cord injury. So trunk control is impaired in people with spinal cord injury, and restoring volitional control of trunk musculature can enable recovery of gait and balance. So most of the current work that has been done has focused on utilizing neuroprosthetics for upper limb rehab and of late lower limb paralysis, but there is still a lot of work that needs to be done to enhance volitional control of trunk. Okay, so the recent work that has been done in the field of neuroprosthetics has been the development of brain-spinal interfaces. The idea here is to record the movement intent from the brain, decode the movement intent, and stimulate the spinal cord below the lesion level and with epidurally and restore volitional control of walking. So recently, this has been translated into a human study in which a patient who had a incomplete spinal cord injury at the cervical level was able to voluntarily step and walk. This is a remarkable achievement, but there is still a long way for us to go. So in this study, called the brain-spinal interface STIMO study, initially, they put the patient to 5 months of rehabilitation, where the patients just received epidural stimulation for 5 months. And after that, the patients received 36 months of home stimulation, where the epidural stimulation was like, the patient can use it whenever they need. And post that, there was an implantation phase where ECOG implants were implanted on the patient, and the patient was allowed to recover, and the patient underwent 40 sessions of neurorehabilitation with this brain-spinal interface. And post the brain-spinal interface, again, there was the home use part of the study. And by the end of the BSI STIMO study, the patient was able to voluntarily get up and voluntarily make a step and make more adaptive movements, and was able to walk naturally. What was even surprising was that during the stim off condition, when the stimulation was off, the patient was able to make remarkable improvements in the distance that the patient was able to walk in 6 minutes. So here you can see that initially, with just the stimulation on, without the brain-spinal interface, initially the performance was poor, and then slowly it reached a plateau. And after the BSI treatment, and even under the stim-off condition, the patient was able to remarkably cover more distance within the 6 minutes. And the balance was also, the balance under the stim-off condition, post-BSI compared to the pre-BSI was improved, and also the gait was higher. So what this means is that the brain-spinal interface is not only acting as the bridge that bypasses the lesion site, but it's also enabling neuroplasticity by strengthening the spared circuits that existed, allowing for the improvement of gait and posture, even when the device was actually turned off. It's cervical. It's cervical, C5, incomplete. They were all incomplete injuries. Yeah.
I actually saw this individual. I wouldn't say it was walking naturally.
Okay.
I would say that the people in the similar trial, if you look at how they're walking, they're using a sort of a reverse motor
Okay.
So he was had better ability to, modulate the height.
Right.
The second was I wouldn't say he just lost walking height.
Okay. 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 So
since we're here, like, what's the improvement on the 6-minute walk test? What's the maximum? Like, what's the scale? The top 0.
Okay. That
It cut off, but
okay. Cut out.
it's fine. I guess we can, because that's important. When
Yeah.
these are clinically. I've seen the study. I just don't remember off the top of my head.
60.
60. So there was
60.
like a
60 meters.
10-ish meter improvement
Yeah, I would say so. Yeah.
from plateau. So he was roughly maybe 10 meters. Yeah, so that's like overall. Okay.
So, Bharadwaj, then it raises a question, right? Like, how long does that effect last? So if you were to kind of continue beyond 48 minutes, like, how long would that last, or does it come down, and
Yeah,
how
that's-
far does it fall, right?
That's an interesting question. We don't know the answer to that. Yeah. How long does it last? Yeah, it'll be an interesting follow-up to do, to really see whether it's spared
Yeah.
neural circuits that actually enabling this.
I mean,
Yeah.
this result by itself is super interesting, right?
Yeah,
Like,
it's all under
it's very
stimulation,
interesting.
off stimulation. That is the fascinating part.
Yeah.
Nevertheless, to really get off the wheelchair, he's still having, if you look at it, he's still having support. He's still having some assistance, crutch. He's able to stand up. He's able to initiate weight-bearing steps, but he still has the walker. Right? To really get rid of the walker, what we have to really understand is how can we control the lateral stability of posture. So if you see all of these studies, that's where they're doing this brain-spinal interface for lower limb paralysis, all of them, from rats to humans, all of them, they're on a body weight support, which means some of the trunk control is like taken away, and the lateral stability is like taken away. So to really get at it, we have to understand what are the brain circuits that encode for lateral stability. And it turns out that the whole of the central nervous system participates in the neuronal control of posture. So you have spinal reflexes that are short latency responses that are non-functional, and then you have these reflexes that are medium latency, and there are these brainstem synergies that can kick in during unexpected postural perturbation. And then you have we also have the cerebellum that can adapt to experience, and basal ganglia that does action selection and is involved in more complex situations. And then you have the long latency cortical response that's involved in stepping and reaching in uneven terrain. So the entire nervous system participates, and the primary sensory signal from the posture is coming from the load and the position afferents in the extremities, and also in the visual and vestibular signals coming from the visual and the vestibular system. And in humans, primarily the proprioceptive afferents play an important role in postural correction. So the question is, can the cerebral cortex be a good source for decoding postural corrections? It's easy. It's easy because a lot of the other studies it's easy to access. A lot of the other people are accessing it. What if we can understand how the cerebral cortex can encode for postural perturbations and use the cortex as the readout for encoding for postural corrections? So it turns out that previous research has shown that the trunk motor cortex is plasticity is very necessary for the recovery of stepping in complete spinal cord injury models. So this is a study in the rodent model. So what you're seeing is is a ratunculus, which is like the rat version of the homunculus, with the more medial portions representing the motor cortex of the rat, that's like dotted, and the more lateral portions showing the somatosensory cortex. So if you take a look at this map, you can see the barrels are very well defined in the somatosensory. The forelimbs, these are the digits, that's very well defined. Hind limbs, digits very well defined. But you don't see any information on the trunk here. And also in the motor cortex, you don't, it's a very crude map. And we can get this map by actually sticking an electrode in this part and actually stimulating the brain and recording muscle activity and movements, and then you can categorize saying, oh, a trunk muscle moved, or trunk muscle got activated, so that's trunk motor cortex. And here, you get to get the recording part, so you can stick an electrode here and touch the body and look at the neurons that respond, and then identify, okay, that's trunk's sensory information getting processed. So that's how you identify it. And we looked at the previous research, they looked at these maps in completely spinalized animals, and without any therapy, they found that the hind limb portion of the trunk motor cortex was completely silent. Makes sense because you have a complete cut. And because the animal was walking around a lot more with its forelimb, to compensate for that, the forelimb motor cortex became more responsive to forelimb somatosensory input. When the animals got a combination of therapy of treadmill, bike exercise, and of serotonergic agonists, there was widespread reorganization in the trunk motor cortex in a manner that was beneficial to the improvement in the recovery of weight-supported stepping. So there were 3 groups in the study. So the sham therapy group, the animals barely recovered weight-supported steps. Partial therapy was bike and 5-HT, and there was a modest recovery of weight-supported steps. But in the animals that got treadmill, bike, and 5-HT, there was a good amount of recovery in the weight-supported steps. And this amount of recovery paralleled the expansion of the trunk motor cortex into the deafferented hind limb motor cortex. So what's going on is that the axatomized hind limb axons are now, instead of dying back, they're now sprouting above the level of injury and innervating the trunk musculature. So that's how you get the trunk motor cortex expansion. And if you lesion this reorganized area of the trunk motor cortex, you lost all the benefits that you got through the therapy. So this suggests that the trunk motor cortex is not only is an essential part of the recovery process. And this suggests that this could be a good source where we could study the neuronal encoding of posture. But if you see these maps, we don't see a lot of detail here. So as part of my doctoral work, I followed up and tried to revisit and understand the organization of the trunk sensory motor cortex. So to envision a postural BMI, we need to identify a good source and detect the falling signal, and then use that as a control signal to a neurostimulator to say, let's say, extend or flex a leg. So the building blocks for a postural BMI is the following. The first is to examine the organization of the trunk motor cortex. Second is to understand sensory motor integration. Then to understand the cortical computations underlying postural control and how these systems are affected by spinal injury. And finally, what is the effect of physical rehabilitation after spinal cord injury on these underlying computations and the recovery of posture and gait. So to examine the organization of the trunk motor cortex in the rodent, I implanted EMG electrodes on the forelimb, parts of the trunk, upper thoracic, mid thoracic, lower thoracic, and the hind limb. And basically, we would stimulate the brain and record movement and muscle activity all over the body. And this way we could examine the organization of the trunk motor cortex, how much of it is involved in controlling forelimb and hind limb. We could study what are the overlaps, and we could also now dissect out, okay, are there different regions in the brain that control upper, mid, and lower thoracic trunk musculature. Then, to understand sensory motor integration, we can insert recording electrodes in the trunk motor cortex and stimulate the periphery. We can stimulate the forelimb, hind limb, upper trunk, mid trunk, and record the sensory evoked potential. And also, we can estimate current source density, and we can do current source density analysis that will basically tell us where and when along the cortical column is the synaptic integration happening, to understand where the underlying source of the sensory input is coming from. So this is a probability distribution map across multiple animals. And if this dot is like white, this means that like in every animal that I went to and stimulated that part, that was a trunk movement. So for example, all of these black spots, we did not find any trunk movement. So this is the boundary of trunk. And what was interesting to us was like the trunk motor cortex was huge, but most of the trunk representation was dedicated to overlapping representation of the hind limb, which was more caudal, and the overlap with the forelimb was smaller and it was more rostral. They're more likely to be polysynaptic because in the rodent, there are very few monosynaptic connections from the brain. So more likely to be, polysynaptic. And I looked at it, I did not really see any differences in timing, or I think my EMGs were not robust enough to really get at the timing aspect. Yeah. So trunk M1 extensively coactivates with hind limb, more than forelimb musculature. And within trunk M1, when we looked at the conditional likelihood to activate lower thoracic or upper thoracic, there was a clear somototopy, with lower thoracic trunk motor cortex being more caudal and medial, and upper thoracic trunk motor cortex being more rostral and lateral. So across the entire caudal rostral extent, there is a relationship, which means that there is an internal somototopic organization of motor output that exists within trunk M1. So next we inserted recording electrodes, stimulated the body parts to understand sensory motor integration in trunk M1. And we examined the sensory evoked potential that you see here. And surprisingly, we found that across the entire rostrocaudal extent, in both the regions spanning lower trunk and upper trunk, hind limb somatosensory information was the highest, more than even trunk itself. So what this suggests is that like the sensory motor integration in trunk M1 is homototopic, is not homototopic, and is rather functional. This was very unique compared to other systems. For example, in forelimb sensory information the integration between forelimb sensory and forelimb motor integration is highest compared to the other body parts. Hind limb also is the same thing. But it seems like for trunk motor cortex cared about where the hind limb sensory information more than even trunk in itself. And it makes sense, and this could be a useful circuit for maintaining balance. So to do that, we actually did a control experiment where we went to the receptive field center in hind limb cortex, forelimb cortex, and trunk cortex, and we changed the current from light 0.5 milliamps to 5 milliamps, and we saw that the responses at the receptive field centers were comparable at all the stimulation currents. So that kind of told us that it's whatever we are stimulating is the equivalent response to the brain. the responses, the evoked responses at the center of the receptive field to trunk, at the center of trunk sensory cortex, center of forelimb sensory cortex, center of hindlimb sensory cortex. The evoked responses were similar at all amplitudes. So that kind of told us that it's not a difference in receptor density at the periphery, and we are kind of comparing equal amounts. Yeah. Yeah. Okay. When we looked at the current source density analysis, in the trunk motor cortex, the hind limbs, in response to hind limb stimuli, the sensory input arrived earlier than the response to trunk stimuli. So trunk M1 receives hind limb somatosensory input earlier than trunk sensory input. So this also kind of suggests us that maybe the trunk hind limb sensory information is more relevant to the trunk motor cortex. And then to follow it up, we did some tracing studies with the help of Dr. Francois Poisson, who was a postdoc at Karen's lab, and we injected a retrobead tracer in trunk M1, and we looked at the connections. And we found that in all the animals, we could see strong labeling in the VPL portion of the thalamus, suggesting that probably it's the proprioceptive input from the hind limb is directly coming through this VPL to M1 connection. Of course, we have to do more follow-up studies, but this is our likely hypothesis of how trunk M1 receives hind limb somatosensory. Yeah, yeah, yeah. So the conclusion we drew from it was that hind limb stimuli was more relevant for trunk M1 activity, and this sensory motor integration could be useful for maintaining balance. Now, until this point, all we have done was anesthetized experiments. So to really understand the encoding of postural control, we built an experimental setup where animals would receive unexpected postural perturbation, basically 4 tilt types. It would either receive a slow clockwise tilt, a fast clockwise tilt, slow counterclockwise tilt, or a fast counterclockwise tilt. And while the animal was doing the task, we can record ground reaction forces from the platform, and we can simultaneously also record neural activity from the trunk somatosensory cortex, parts of the forelimb somatosensory cortex and hind limb somatosensory cortex, and also parts of the lower trunk and upper trunk and hind limb motor cortex. So to understand how neurons are encoding information, what we could do is we would do 100 tilts of each type, and then ask on a real-time basis, can I classify how on a single trial basis can I predict the tilt type of what happened? We could do this more both at the single neuron level and also the population level. So basically what we can do is we can do 100 tilts of each type and look at the neural activity and build a template. So these are 4 different templates, and then we can tilt the animal once and in real time classify whether the single trial activity belonged to either one of the tilt types, and classify and estimate the amount of information encoded in the brain about that particular tilt. So if it's 100% correct, it's 2 bits of information. And when we quantified the amount of information present in the brain about the postural, about the tilt, we found that the motor cortex has more information about the brain, about the tilt that happened more than the somatosensory cortex. And when we looked at the population as such, we found that, again, motor cortex conveyed more information, and even when we added the neuronal activity from the somatosensory cortex, there was no big increase, suggesting that there is a lot of redundant information in somatosensory cortex, and motor cortex is probably involved in higher order processing related to trunk stabilization that happens during the postural control. So this is a response of a neuron to a single tilt. So this is so here you see in dotted lines is the fast tilt and the slow tilt, and you can see the same neuron responds differently for both the tilt types. For the fast tilt, it fires a lot during the initial phase of the perturbation and then slows down and then stops firing. The slow tilt, it fires and then scales throughout the perturbation. And we can study these responses in the different regions in the brain that we implanted, and we saw, like in the somatosensory cortex, the proportion of neurons that responded to tilts were higher in the hind limb compared to the forelimb, and they were the least in the trunk somatosensory cortex. This is likely because the trunk somatosensory cortex is not getting a lot of proprioceptive input from the forelimb and the hind limb, and that's why it's low. And when we looked at, within the trunk cortex, when we looked at the subdivisions of lower thoracic trunk sensory, mid thoracic trunk sensory, upper thoracic trunk sensory, they were all like really low and not very different. And within the motor cortex, both the hind limb motor cortex and the trunk motor cortex were equally likely to respond to tilts. And within the trunk motor cortex, though both the upper thoracic and the lower thoracic were equally likely to respond to tilts, the lower thoracic part of the motor cortex conveyed more information about the tilts that occurred than the upper thoracic trunk motor cortex. So this suggested that, this kind of tells us that the lower thoracic portion of the trunk motor cortex is more specialized in the neuronal encoding of posture. And this could be a useful site where we could record, where we could decode information about postural control of lateral stability. So overall, in the first study, we show that the sensory motor integration in the rodent trunk cortex is dominated by hind limb sensory information, and this sensory input somatosensory input is likely to be proprioceptive. And we show that the lower thoracic trunk motor cortex is specialized in the neuronal encoding of posture. And in this paper, there is also more details about the organization of the trunk somatosensory cortex, how the dermatomes are organized at the spinal level, and also how the dermatomes are organized within the trunk somatosensory cortex. So this work was led by Dr. Gary Blumenthal from our lab. And if you're interested in the trunk somatosensory cortex, please check this paper out. Okay, so till now we were looking at how single neurons were encoding for postural control, and looking at single neuron activity sometimes is very noisy, and it can be stochastic when trying to relate to behavior. So instead of looking at single neuron activity, when you look at multiple neurons together at a time, some patterns start to emerge. So we can do some dimensionality reduction techniques and uncover these patterns, and this is called a latent state. So here you see like a three-dimensional neuron activity is now simplified into a two-dimensional plane. This happens because neurons are often interconnected with each other, and they live in a network, and there is a lot of redundancy in how neurons actually fire. So we don't have to look at every neuron. All we have to understand is what is the low dimensional feature that will explain the behavior that we are trying to measure. So we did principal component analysis, which was the simplest dimensionality reduction technique, and we saw that surprisingly, the first three principal components explained at least about 60% of the variance in the data. And then we saw three types of these latent neural activity emerge. We saw an excited component, which basically scales to the speed of the task and did not care about the direction in which the tilt happened. And then we saw a mixed component where it would it was purely sensitive to the direction of the tilt, but would not care about the speed. And then we saw an inhibitory component, which was more of a detection mechanism. All it cared was whether a tilt happened or not. And this latent dynamics were pretty consistent from animal to animal, and it was very robust. So this was very encouraging, because then we could like look at these latent dynamics and then look at how they are encoding for behavior. Okay. So when we looked at the single neuron level to see whether these types of dynamics were represented in the single neuron level, we looked at them and we saw the same types emerge. So we classified them into excited neuron, mixed neuron, inhibitory neuron, and then we looked at the proportion of their cell types in the different regions in the hind limb motor cortex, trunk motor cortex, hind limb somatosensory, trunk sensory, and forelimb sensory. And what we saw was like in the trunk somatosensory cortex, we found a large proportion of these inhibitory neurons. And this was very interesting, because what it told us was that trunk somatosensory cortex cared about whether a tilt happened or not, but it was not good in encoding for what type of postural perturbation exactly happened. And we saw that these these different neuron types were functional. For example, the excited neurons were better at decoding speed than the mixed ones, and the mixed ones were better at encoding direction, and the inhibitory ones would just not care between distinguishing between speed or direction. All it would care was whether a tilt happened or not. So the trunk neurons mainly encode for three things: speed, direction of tilt, and the direction of postural perturbation. So next thing is, can we use these neuronal dynamics to decode behavior in real time? And are these neural dynamics, do they still exist after spinal cord injury? So this neuronal dynamics work was spearheaded by MD PhD student who joined our lab, and this is Greg Desai, and he brought in all the computational expertise and showed that when you look at behaviorally relevant dynamics, when the animal tilted to the right and left, you had these non-overlapping trajectories that existed. And what this meant is that when the animal is actually tilting, it's going to flex on one limb and it's going to extend the other. So to the left, and when it tilted to the right, the other thing is going to happen. So the idea that the trajectories were not overlapping suggested that there is very less chance for the cortex to flex both the legs at the same time. So in stroke and in other neurological diseases, a lot of the times the synergies get merged. And here the cortex was doing this, keeping this flexion extension of both the limbs in separate latent subspaces. And what he showed was like we could also use these dynamics to decode behavior of the ground reaction forces in real time. So you can check out our paper that just got published recently. So next part is, what is the role of physical rehabilitation? So to do this, I looked at an incomplete spinal cord injury in the rodent model. It's a moderate contusion at T10, and the animals received physical rehabilitation for 5 weeks. And we saw that like the animals that received exercise therapy were able to take more frequent weight supported steps and had better interlimb coordination. And then we took these animals that got the therapy and non-therapy, and we let them walk on a glass plate, and we looked at the base of support between the hind paw. So what this is just the distance between the two hind limbs. So we saw that like in the no therapy animals, to be able to walk well, the base of support like was wider in the animals that did not have therapy. So this was a kind of compensatory strategy of the animal to balance itself as it walked across the glass plate. In the therapy animals, surprisingly, you don't see that. So the therapy animals, the base of support remained narrow throughout the entire time. So this suggested that the physical rehabilitation is improving the functional recovery of gait and balance during overground locomotion. Also, we did half an hour of half an hour of treadmill therapy with repetitive somatosensory feedback to the hind limb. No body weight support. We would allow the animal to fall as much as we can so that the animal can pick itself. So these are not like so a lot of the animals have the ability to support weight. So we do that for 5 days a week for 5 weeks. Yeah. It's half an hour per session. And the therapy would start like a week after spinal injury.
So just to follow up on the therapy, you also provided some other sensory feedback systems. How did you provide this somatosensory?
We would just touch the paw, rhythmically touch the paw,
Okay.
and give some somatosensory feedback. There was no machine or anything.
Okay. So no modulation.
No modulation of any kind. It's just tactile touch. We just, yeah. Yeah, touching the paw. Yes. Right. For the vagus, yeah, that's what he does for the vagus now. Yeah. So we did this, and when we looked at the trunk motor cortex in these groups, so this is the naive motor cortex in normal animals. So here is the regions in the brain that activate lower trunk, this is upper trunk, and this is all hind limb motor cortex. So in both groups, what you can see is the trunk motor cortex, regardless of the fact that the animal got therapy, the trunk motor cortex expanded into the deafferented hind limb motor cortex. But what was interesting is in the animals that got the therapy, in the lower thoracic trunk motor cortex, these regions were more likely to activate trunk compared to the animals that did not have therapy. And when we looked at the trunk, the likelihood to activate trunk in the lower thoracic trunk motor cortex on a per animal basis, again, we saw that the exercise therapy animals had a bigger likelihood to activate trunk in the lower thoracic trunk motor cortex. And when we correlated that to the functional, the BBB score in the open field, we saw that there was a correlation of the likelihood to activate trunk in the lower thoracic part of the motor cortex. And then we also looked at the EMGs in the trunk muscles, in the lower thoracic longissimus, the dorsal muscle, and the lower thoracic oblique muscles, and we found that exercise animals had modest increase in the EMG amplitude compared to the non-exercise animal when you stimulated this lower thoracic trunk motor cortex. And surprisingly, we found that the non-exercise animals tend to have a higher EMG amplitude in the thoracic longissimus compared to the exercise animal. What this kind of tells us is like if the animal, because the injury was moderate, there's going to be a lot of spontaneous recovery. And the brain is kind of figuring out strategies to relearn motor control to enable walking again. And how it's doing that is very different. In the non-exercise animal, it's kind of activating the mid-thoracic longissimus muscle, the dorsal muscle. It's stiffening itself up, and it's putting more weight on the upper body and walking. And in the exercise therapy muscle, in the exercise therapy group, instead of putting more weight on the upper body and stiffening the trunk, the dorsal trunk, it's now activating the obliques below the level of the lesion, and it's walking. And that is more similar to how a normal animal would walk. So, Yeah, so yeah, you bring up a good point. We were thinking of that too. We looked at the spinal cord between the two groups, and we looked at this amount of spared matter. I think I have that slide. Sorry, a lot of backup slides. Yeah, and we were doing typical cutting, staining, and quantifying the white matter tracks. Maybe like with light sheet microscopy, we have more. Maybe it's a resolution. It could also be a resolution issue that but we did not see differences in the amount of. Yeah, so when I started out working on spinal cord injury, I was working on neuropathic pain development. So we would give the animals the same at T10. We have a paper on that. Like we'd give the same injury across animals, but then the likelihood of developing at level pain at the trunk, or above level pain or below level pain, were very different. And we were trying to study pain at the trunk. So like there was no. We would give the same injury, but the animals would. The phenotype was very different. So I think in contusion injury models, it's very hard to control, to have a tight control. But that's the beauty of it. Like it's more clinically relevant. It's more real. Yeah. Okay. So in these exercise animals, we also looked at how are they now responding to postural changes, and we looked at the information, the amount of information in the trunk motor cortex, and we saw that like if the animal got no therapy, the information conveying neurons will drop by 2 months after injury, but when the animal had therapy, somehow the information conveying neurons would not drop. They would stay the same, but the amount of information that they conveyed about the postural task dropped after a spinal cord injury. And this was not different between the two groups. So physical rehabilitation preserves neurons that convey information relevant to postural changes. Somehow activity-dependent plasticity is strengthening those spared neurons and preventing them from dying. But again, they lose their functional ability to code for the different postural changes. So this is still like work in progress, and yeah, we are still preparing the manuscript. And what was really surprising to us is that like 2 months post-injury, the neuronal dynamics were so stable in the trunk motor cortex that we could decode the changes in the platform angle 2 months post-injury, and they were similar. So this kind of suggests that there are like some parallel, if the lower limb is lost, there are some parallel streams of computation that's going to the brain and still informing the motor cortex about about postural changes. And so this is very good and promising, which means that trunk motor cortex can be used as a source where we could decode changes in postural stability even months after spinal cord injury. So the key takeaways are that we showed that the somatotopic organization of the trunk motor cortex exists with extensive preferential integration of hind limb somatosensory input, and this is useful for maintaining balance. We also show that the cortical representation of the lower thoracic trunk motor cortex is specialized in the neuronal encoding of posture, and rehabilitation in fact enhances this cortical control of lower thoracic muscles below the injury level. And finally, physical rehabilitation preserves the neuronal information relevant to postural control, and the cortex can continue to encode for postural stability months after spinal cord injury. And this is my pet hypothesis of how this could happen. And the hypothesis is that like in exercise animals, the corticospinal tract is more likely to sprout to the cervical, sprout into the cervical enlargement, And there you have these long descending propriospinal axons that could like establish direct control of lower thoracic trunk muscles below the injury level. The other surprising things that we saw is that the likelihood to exclusively activate lower thoracic muscles was much higher in the exercise group compared to the non-exercise group. So in the non-exercise group, when we saw lower thoracic trunk lower thoracic muscle activation, it was most likely to co-activate with the mid-thoracic muscle. So in the non-exercise group, we think that maybe the LTM1 axons sprout a rostral to the lesion, and maybe there is some weak co-activation that still exists because the trunk is a biomechanical couple and there's a lot of sparring, and that's how we're able to activate the lower thoracic trunk muscles. Of course, to test this hypothesis, we'll have to do tracing studies to really confirm whether that's true. These are rats, right? Right. Yeah, so it's been kind of done. So there's been work by Corteen group where they show that there is this cortico-reticulospinal reorganization. So with the severe contusion, the reticulospinal neurons that are very lateral on the spinal cord, they are somehow spared, and somehow the cortex gets access to areas below the injury by sprouting to the vestibulo VGI nucleus in the brainstem and gains access to these reticulospinal tracts, thereby establishing this indirect route. And yeah. Mm-hmm. Yeah, so we tilted. Okay, I see. We tilted 18 degrees, and we tilted. The tilt paradigm that we designed was such that the likelihood for the animal to lose complete balance was not there. That's a good point. You raise a good point. How will the brain then behave, right? Yeah, you're right. Yeah, we did not scale it up. That's a good point. Yeah, and lastly, I'd like to thank all my mentors who made it possible for me to be here. And so this is my journey. I moved to the US 2013, went to Drexel, and I was introduced to neuroscience. I was an electrical engineer, and I was introduced to neuroscience by Dr. Karen Moxon. And she introduced me to the world of EFIS, neuro rehab, spinal cord injury, and I stayed along with her. And Dr. John Bethea too. I did some collaboration with him on neuropathic pain development, and he's been kind of a co-mentor to me. What is it? Yeah. Yeah, and then I stayed on with Karen, went to UC Davis. I had to move during my PhD to UC Davis, started again, graduated during COVID. So this is, I did my entire graduation on Zoom. So this is kind of my podium talk. So I'm lucky, and it's exactly been a year since I joined Miami Project. I joined October 2nd, 2023, and it's nice. And Patrick has been a great mentor to me, and I really love being part of the Miami Project, and looking forward to do some great science while I'm here, and hopefully push to the next step where I become independent at some point. And yeah, I spent 2 years doing a postdoc at the Neuromodulation Research Center, where I was evaluating pathophysiology of sleep dysfunction underlying Parkinson's disease in non-human primate models. Worked there for 2 years under the mentorship of Dr. Jerry Vitek, and Dr. Luke Johnson. And yeah, now I'm here back with Patrick. We had the same PhD advisor, and yeah, It's nice to be here. Thank you.